Retatrutide resulted in body weight loss of up to 28 to 30% of body weight at 104 weeks in the TRIUMPH-1 trial, as well as blood pressure decreases. In addition to that, we see increases in your good cholesterol and your HDL, while decreasing your bad cholesterol, your LDL. And we also see fasting blood sugar goes down, but there's a few side effects to that, being increases in nausea and maybe vomiting, depending on the dose, as well as an elevated heart rate. Would you take it given that? This is Retatrutide. Today we're going to be talking about how it works. We're going to talk about its mechanism. We're going to look at the evidence base and see what it suggests, and we're going to look at any risks associated with it. So if that all interests you, please come along with me on the video today and we will get right into it. So Retatrutide is a 39 amino acid peptide, and a peptide is basically just a chain of amino acids linked together. And it's been deliberately engineered so that different parts of that chain can activate these three different hormone receptors. So the first region, it interacts with the GIP receptor. The middle region interacts with the GLP-1 receptor. And another region interacts with the glucagon receptor, or GCGR. So that's why Retatrutide is, in practical terms, described as this, triple receptor agonist. And when I say agonist, I just mean a molecule that binds to a receptor and then activates that receptor. And so kind of in practical terms here, it just combines three hormonal signals in this one molecule. So the GIP and GLP-1 components, they support this glucose-dependent insulin secretion and nutrient regulation. And again, we'll get into this more in the video later, but the glucagon receptor component then increases energy expenditure and actually also promotes kind of the movement of stored fuels. So we'll look at fatty liver and how glucagon receptor can, when it's activated, can affect that. So there's also this fatty acid, this fatty diacid, so 2, this fatty diacid side chain, and it's attached to the peptide through this chemical tether. So this fatty chain, it can bind reversibly to something called albumin, and albumin is this big protein circulating in the blood, and it basically acts like this temporary carrier, or you can think of it as a reservoir, and all that does is it slows the drug's clearance and protects it from rapid degradation. So if you just had the Retatrutide peptide without that fatty acid side chain or this albumin binding component, it might be degraded in minutes, compared to when you have it here, you could have something that was more along the lines of a once weekly injection. Now, the amino acid side chain also does contain several kind of unusual modifications here, but the chemists did design this, and it has this AIB, or alpha amino isobutyric acid, as well as this modified leucine. And all these changes do is it basically helps make the peptide more resistant to break down by enzymes. So rather than being chopped up extremely quickly like a normal gut hormone, it remains intact for longer. So both these side chain little pieces there, as well as the fatty side chain tether, they all kind of work together to prevent this quick degradation. And that long duration of action here is what allows Retatrutide to be investigated as a once weekly subcutaneous injection. So when I say subcutaneous, I just mean an injection into this fatty tissue underneath the skin rather than directly into muscle or a vein, say. And so the big picture here is that Retatrutide is this engineered peptide designed to activate the GIP, GLP-1, and glucagon receptors, while its amino acid modifications and albumin binding fatty side chain then allow it to stay active much longer than the natural hormones. And so it's injected typically, again, subcutaneous once a week, and the phase 3 studies are also evaluating once weekly dosing. The sponsor of today's video is Peptavo. Peptavo is an end-to-end peptide therapy course run by Dr. Jesse Morse, a board-certified physician who has been prescribing these compounds in an active practice for 7 years. There are 12 modules and 60-plus peptides covering mechanism, dosing, contraindications, and monitoring, with every protocol graded on how strong the evidence behind it actually is. It's built for licensed clinicians like physicians, nurse practitioners, and physician assistants. Module 1 is free to preview first, and it's also worth knowing that they're going through CME accreditation right now, and the price goes up once that lands. So peptavo.com. Okay, so now let's talk about the action of each of the different parts of Retatrutide. And instead of talking right away about GLP-1, so things like, Ozempic or, Wegovy, why don't we start with GIP, okay? So GIP, think tirzepatide. So tirzepatide activates the GIP receptor. And so GIP stands for glucose-dependent insulinotropic peptide. And GIP is one of the body's incretin hormones. And all that means is that it's a hormone that's released by the gut after you eat. And it basically helps prepare the body to deal with the incoming nutrients. And more specifically, GIP is released by specialized cells in the small intestine called these K cells. So as soon as food, especially carbohydrates and fats, enter the small intestine, these K cells secrete GIP into the bloodstream. Now, GIP then travels to the pancreas, where it acts pretty much on the beta cells of the pancreas. And these are the cells that produce and release insulin. And insulin is the hormone that lowers your blood glucose by helping tissues take up glucose from the bloodstream. So just to kind of put that together, GIP, it binds to the GIP receptor on the surface of the beta cell. And this receptor is what's called a G protein coupled receptor. You see that kind of turquoise little curly thing on the cell there. And GPCRs, I'm going to call them GPCRs from now on, they're this kind of huge family of receptors that sit in the cell membrane. And so instead of directly changing what happens inside the cell, they activate proteins called G proteins, which then start this kind of intracellular signaling pathway. So there's this secondary message that happens. And so if we kind of walk through these steps here, in this case, the GIP receptor activates a G alpha S protein. So the GS basically stands for stimulatory because its job is to stimulate the enzyme adenylyl cyclase. And adenylyl cyclase then converts ATP into cyclic AMP, or CAMP. I'm going to call it CAMP from now on. So as a result of all this, the concentration of CAMP inside the beta cell increases. And just like we're going to talk about later, CAMP is this secondary messenger. So it's basically the molecule that carries the signal from the cell surface deeper into the cell, and it kicks off another signaling cascade. So that's why, in biochemistry, CAMP is referred to as a secondary messenger. And so once these CAMP levels go up, it activates these two really important signaling proteins, protein kinase A, or PKA, and EPAC2. And that stands for exchange protein activated by CAMP2. And so PKA is something called a kinase, and it works by phosphorylating different proteins inside the beta cell. And when I say phosphorylate, that's just a fancy way for saying it attaches a phosphate group it attaches this phosphate group onto a protein inside the beta cell. And so by doing that, it changes their activity. And EPAC2, it works a little bit differently. So rather than phosphorylating proteins, it helps regulate proteins involved in moving insulin-containing vesicles toward the cell membrane. So a vesicle is just, you can think of it like a soap bubble, and it contains insulin inside this soap bubble. So although PKA and EPAC2 use these different mechanisms, they both make the beta cell much more efficient at releasing insulin. Okay, so let's keep moving here. So here's the really important point. So GIP doesn't force the pancreas to release insulin all the time. Instead, it kind of amplifies insulin secretion only when blood glucose levels are already elevated. So that's why you'll hear that GIP stimulates glucose-dependent insulin secretion. And this is actually a really smart safety mechanism. So if your blood glucose is, say, normal or low, GIP has very little effect on insulin release. But then, after you've eaten, when glucose levels rise, GIP makes the beta cells respond much more strongly, so they actually release a larger amount of insulin than they normally would. So that's actually known as the incretin effect. So if you drink, say, a glucose solution, you drink this, think of it like a big Gatorade or sugary drink, and compare that to receiving the exact same amount of glucose directly into a vein, your body releases much more insulin when the glucose passes through the gut. Okay, so hopefully that's helpful in kind of working through that. So there's this other thing called DPP4. If we look at the bottom left there, this is dipeptidyl peptidase 4, and it's this enzyme, and it's found throughout the body. And basically what it does is it rapidly breaks down these incretin hormones like GIP. So GIP only remains active for a few minutes before DPP4 inactivates it. So this it's a different conversation, but there's also these things called DPP-4 inhibitors, and they're this class of medication that's used to treat type 2 diabetes, and they work by actually preventing this breakdown of DPP-4. But I just want you to keep in mind that DPP-4 is always there, and it's looking to, chop up GIP or GLP-1 or say, a glucagon type peptide. So it's looking to chop those up quickly. Now, when you have binding to albumin, it's harder for DPP-4 to then chop up these kind of stimulatory peptides. So, okay, let's just bring this back together here. So the big picture here is after you eat, GIP is then released from K cells in the small intestine, and then it moves over to the beta cells in the pancreas, and then it binds to this G protein coupled receptor. It activates something called this GS protein that increases cyclic AMP, and then it activates PKA and EPAC2, and then together, these signaling molecules, or these signaling pathways, make the beta cells much better at releasing insulin, but then only when blood glucose is already elevated. And at the same time, GIP can also promote fat storage in adipose tissue and does have some modest effects on glucagon secretion, and it's also rapidly broken down by the enzyme DPP-4. Okay, so that was kind of a big overarching view of what happens when, say, the GIP component of retatrutide binds to a GIP receptor on the beta cell in the pancreas. So these following ones, when you go through GLP-1 and glucagon, will go a little bit quicker because a lot of these cascades are somewhat similar within the cell. So now if we move over to GLP-1, GLP-1 stands for glucagon-like peptide 1, and just like GIP, GLP-1 is also an incretin hormone. And when I say incretin hormone, I basically just mean that it's released from the gut after you eat, and it basically just helps the body prepare for the incoming nutrients. And so the difference here, though, is where it comes from. So this is why GLP-1 is quite different from GIP. So GLP-1 is released by the L cells, which are found mainly in the lower part of the small intestine, as well as the colon. And once it's released into the bloodstream, one of its main targets is the pancreatic beta cell. So similar to GIP, it also has a target at the pancreatic beta cell. And then GLP-1 binds to the GLP-1 receptor, which, just like the GIP receptor, is also something called this G protein coupled receptor, or GPCR. Okay, and then we have the same kind of cascade here. So, we activate the G-alpha S protein that stimulates cyclic AMP, and then we have that secondary messaging where we have the increase of cyclic AMP, and then as a result of that, we get deeper into the cell, PKA increases and EPAC2 increases, and then together these proteins make the beta cell much more likely to release insulin. And again, just like GIP, it's a glucose-dependent insulin release. So you need to have glucose in the bloodstream for this insulin to be secreted when we have this specific cascade going on. So GLP-1 is quite similar in its effect at the pancreatic beta cell. Now, it also has some things that are maybe a little bit different than GIP. So GLP-1 has these several additional effects outside the pancreas. And, this is really what makes it such an important target here and interest to a lot of these companies that are looking at developing weight loss molecules. Because first, GLP-1, it also acts on the pancreatic alpha cells, where it reduces glucagon secretion. So glucagon, it normally tells the liver to release glucose into the bloodstream, which then lowers, this lowering glucagon helps reduce blood glucose, say, after a meal. Okay, so that's one effect. The second is that GLP-1 acts on the stomach, where it actually slows gastric emptying. So instead of food, say, moving quickly into the small intestine, it leaves the stomach much more slowly. Okay, so that slows glucose absorption, and it just helps prevent large spikes in blood sugar after eating. Okay, so that was the second one there. And then finally, GLP-1 also acts on the brain, and these are particularly like areas involved in appetite regulation. So at the hypothalamus, it could act at the arcuate nucleus. This is important in appetite regulation. And there it increases satiety, which is this feeling of fullness, and it also decreases appetite. Those are kind of two sides of the same coin there. But As a result of this, people naturally tend to eat less, which is why GLP-1 receptor agonists like semaglutide have become so effective for treating obesity and promoting weight loss. And again, just like GIP, you have DPP-4 that wants to quickly come break down GLP-1 when it is in the body there. So that's kind of a similar pathway to GIP, but GLP-1 has a few additional effects that are somewhat unique from GIP. Now we have glucagon. So when blood glucose levels fall, like if you've been, say, fasting overnight, or maybe you haven't eaten for a while, the pancreas, it releases this hormone called glucagon, okay, and specifically the alpha cells in the pancreas. And I want you to think of glucagon's whole job is basically to tell the body, we don't have much glucose around anymore, so let's start using our stored energy instead. That's kind of the message that glucagon sends. Now, glucagon, it doesn't actually enter the cell. So instead, it binds to a receptor on the cell membrane. Again, it's another GPCR type signal here. So another glucagon receptor is another GPCR. And then we have a similar signaling pathway inside the cell. So, we have that pathway that we described earlier, and I've shortened it down a little bit. So we're now just going to skip over to the cyclic AMP part. And then cyclic AMP, again, sends off this whole cascade of things, but here is unique. So I'm just gonna slow down a little bit here. Now, one of the main proteins that cyclic AMP activates is protein kinase A, or PKA. So this is the one that we talked about earlier. But around the same time, another kinase called AMP activated protein kinase, or AMPK, it's also active. And AMPK is basically the cell's fuel gauge. So AMPK turns on whenever the cell is, running low on energy, and it helps shift metabolism towards making more ATP. So this is unique with the glucagon receptor stimulation. So now you've got PKA and AMPK working together. And one of the big things they do is turn acetyl-CoA carboxylase, or ACC, off. They turn it off. And ACC is the enzyme that normally makes something called malonyl-CoA from something called acetyl-CoA. And so I'll simplify this. Malonyl-CoA, it's really important because it's the molecule your cells use to build new fatty acids. So if we think about it, if ACC gets switched off, the cell stops making as much malonyl-CoA. Now, this is really an important step in the whole pathway here, because malonyl-CoA doesn't just help make fat, it also blocks an enzyme called CPT1, or carnitine palmitoyl transferase 1. And CPT1, it basically sits on the outside of the mitochondria, and it acts kind of like a gatekeeper. So these long-chain fatty acids, they can't just walk into the mitochondria on their own. They need this CPT1 to help transport them across the mitochondrial membrane. So when we have malonyl-CoA levels that are high, for example, say after you've eaten, CPT1 is blocked. So it's basically like having the brakes on that. And so as a result of that, fatty acids, they stay out of the mitochondria because the body would rather store fat than burn it. Okay, but now, because glucagon turned ACC off, malonyl-CoA levels fall. And when malonyl-CoA drops, that brake comes off CPT1. So now fatty acids can finally get into the mitochondria. And once they're inside, they go through this process called beta oxidation. And beta oxidation is basically the pathway where long-chain fatty acids get chopped up into basically lots of little 2-carbon pieces called acetyl-CoA. And then along the way, the cell also produces molecules like NADH and FADH2, which carry these high-energy electrons. And then that acetyl-CoA then enters the TCA cycle, also called the citric acid cycle or Krebs cycle, where it's broken down even further. And along this whole way, we're harvesting these electron-carrying molecules. And then the final step at the electron transport chain uses all these molecules that carry lots of electrons to harvest ATP from that, which is then just the cell's main source of usable energy. So all of this whole pathway summed up is basically trying to get energy increasing. So we're switching towards a more energy burning state. And so just to sum that up, glucagon basically, it flips the body's metabolism from, storing fat to burning fat. It increases CAMP, which activates pKa and AMPK. Those enzymes switch off ACC, so malonyl-CoA levels fall, and that removes the inhibition on CPT1, allowing fatty acids into the mitochondria, where they're then broken down through this process called beta oxidation to generate acetyl-CoA and then ultimately ATP. So we're burning energy. And let's just discuss a little bit of the 3-receptor bridge here, because we have all those 3 mechanisms, and now we're going to look at when we have a peptide that works together, how are they going to work together? Okay, so we talked about how an agonist was just a receptor it stimulated a receptor. Typically, in this case, it would be G protein coupled receptors. So we'd have our single retatrutide peptide that would activate these GLP-1 receptors, GIP receptors, and glucagon receptors. And these signals reach different tissues and they overlap, rather than acting as these 3 isolated switches. And so this is important to know because, you might inject retatrutide subcutaneously, but then it's going to have a whole bunch of different mechanisms depending on which part of retatrutide binds to these receptors. So just to kind of sum up what I've been describing earlier, you're going to have all these different pathways that interact, and they're going to influence feeling full. They're going to influence glucose control, specifically glucose-dependent insulin release, and that will affect our glucose response, as well as liver fuel handling. Okay, and when I say liver fuel handling, I just want to bring up the glucagon receptor because there are tons of glucagon receptors on the liver. So that's a specific place where the glucagon part of retatrutide, the glucagon stimulating part of retatrutide, is highly active. So it's highly active at the liver. And just to get back to what we'll get into a little bit later, these human trials, they measured the complete drug. So, it's hard to assign a percentage of weight or glucose change to any one of these receptors. And so you kind of have this holistic effect with retatrutide, and you have some of those effects that we discussed at the start of the video, both the positive effects and the side effects. Now, these receptors, before we move on, these receptors have different potencies. So they activate these, that retatrutide activates these different receptors with different potencies. So researchers have looked at this. So the question that they had was, how well does retatrutide activate each of its 3 target receptors compared to the body's natural hormones? Because, you have all these natural hormones, and then you have this synthetically synthesized retatrutide. So to answer that, researchers use these things called engineered HEC-293 cells, and these are laboratory-grown cells, and they've been modified so each dish expresses just one human receptor. So that's either the GLP-1 receptor, the GIP receptor, or the glucagon receptor. And the researchers, they then added different concentrations of either retatrutide or the corresponding natural hormone. And then we talked about cyclic AMP. They measured how much cyclic AMP the cells produced. So we want to remember here that cyclic AMP, it's that secondary messenger that's generated when these receptors are activated. So more cyclic AMP means more receptor activation here. And the curves on the graph here, they show how the cyclic AMP response changes as the drug concentration increases. So the key number here to remember is the EC50. So that's simply the concentration needed to produce 50% of the maximum response. So a lower EC50 would just mean that it's a more potent agonist because you need less of the molecule to get the same effect. So high means less potent, low EC50 means more potent. And if we look at the results, retatrutide is more potent than native GIP at the GIP receptor. It's slightly less potent than native GLP-1 at the GLP-1 receptor, and it's less potent than native glucagon at the glucagon receptor. So overall here, retatrutide, it activates all three receptors, but it does not activate them equally. It actually has its strongest activity at the GIP receptor. It has this intermediate activity at the GLP-1 receptor, and somewhat comparatively weaker activity at the glucagon receptor. So before retatrutide had its human trials, researchers had the question of which of these three receptors is actually responsible for retatrutide's effects. They kind of wanted to parse out the different effects. And so to start answering that, the researchers turned to mouse studies where they could actually manipulate individual receptors in ways that simply aren't possible here in humans. So what they did for their first experiment is they compared normal, or what you call wild type mice, with mice that had the GLP-1 receptor genetically knocked out. Basically, when I say genetically knocked out, I just mean that they were born without that receptor. And both groups received retatrutide, and the researchers then tracked body weight over time. Now, you might expect that removing the GLP-1 receptor would completely eliminate the drug's effects. But that's not what happened. So the weight loss effect was reduced, but it didn't disappear. So even without GLP-1 receptors, retatrutide still produced weight loss. And the researchers then asked a slightly different question, and that was, what happens if we activate the GIP receptor and the glucagon receptor using these separate selective agonists? You remember, agonist was just a stimulatory molecule. So they combined a selective GIP receptor agonist with a selective glucagon receptor agonist as this physical co-therapy here. So they were administered together. And it's important to point out that this wasn't retatrutide itself. It was actually just two separate drugs given together to mimic part of its mechanism, okay? And next, they expanded the comparison to include several different treatment groups, including vehicle controls, the individual agonists, the combined co-therapy. There was another experimental dual agonist, from what I remember, and there's also this pair-fed control. Okay, so there's a whole bunch of these different comparators that they looked at for these treatment groups. And the pair-fed control one was useful here because, those mice are given the same amount of food eaten by the treated animals. And basically what this does is it just helps separate weight loss caused, simply by eating less from weight loss caused by other metabolic effects. And so here, overall activating multiple receptors produced greater weight loss than just targeting a single receptor alone. So it had this additive effect. And the researchers also measured energy expenditure using this thing called indirect calorimetry. You can just think of it, we're just measuring how much energy was burned, okay? And interestingly here, they actually didn't see a major overall increase in energy expenditure. There was this kind of modest increase during the animal's active or dark phase when the two agonists were combined, compared with the GIP agonist alone, but this overall effect was pretty small. And these experiments, basically what they did here is they kind of help us and these researchers understand the biology in mice prior to studying it in humans. But they don't tell us exactly how much each receptor contributes in humans. And they also don't prove that retatrutide increases energy expenditure in people. So these are all mechanistic studies. So they help explain how the drug might work. But again, the clinical effects still need to be confirmed in human trials, which is where we're going to go next here. So now for the human evidence. So the first in-human study, it gave healthy participants this single subcutaneous injection and then followed serial blood concentrations. So they took a whole bunch of them, and as well as early tolerability. So PK here, it means pharmacokinetics, and that's how drug concentration changes as the body absorbs and clears it. So this small early study oriented kind of the exposure, but not necessarily like the rare safety or chronic efficacy over time. And so when I say efficacy, I mean how well did something actually work in the treatment group? And it's typically done in a lab setting or an experimental setting. So effectiveness is how well something works in the general population. Efficacy is more of like in a research paper, how well did something work? And the concentration curve here declined slowly enough to support weekly development. And this long exposure profile, it kind of helps us explain why these later trials escalated somewhat cautiously, but it didn't establish an approved schedule or what repeated dosing might achieve. Okay, so now we just went over the first study in humans, but now we're going to look at the first full phase 1b clinical trial. Okay, so this was a phase 1b study. It was involving adults with type 2 diabetes, where the participants received once-weekly injections for 12 weeks. So the researchers here, they compared retatrutide with both placebo and something called dulaglutide. And it's this established GLP-1 receptor agonist that's already being used to treat type 2 diabetes. And the goal here wasn't to prove the long-term efficacy yet. It was really just to see whether repeated weekly dosing was safe and to look for some early signals that the drug was working. So this was more of a safety study. So typically, in phase 1 clinical trials, you want to see that a drug is safe, okay? And that's exactly what they found. So compared with both placebo and dulaglutide, retatrutide produced greater reductions in blood glucose and greater weight loss over this 12-week period. And so this study, it basically just established that, repeated weekly dosing was feasible, and it also provided encouraging early evidence that retatrutide could improve both glycemic control and body weight before moving on to these much larger phase 2 trials. Okay, so hopefully that was helpful. They didn't see any of these major safety concerns or side effects. So that was also another thing as well that convinced them that, okay, it's okay to move on to phase 2 now. And the researchers also looked at this other question, and that was, how does retatrutide affect gastric emptying over time? So early in treatment, it actually slowed gastric emptying quite substantially here. So that just means that the food stayed in the stomach longer and then less of the meal emptied into the intestine. But actually, after the patients had been on the drug for a while, that effect became kind of less so. And then the stomach started emptying more normally again. So this is what's meant by gastric attenuation. So all that means is that the stomach, it basically adapts to the drug with repeated dosing. And so this is important because, it suggests that the sustained weight loss seen with retatrutide, it isn't simply just due to the stomach emptying more slowly and forever. So, instead it points towards other mechanisms, maybe more like increased satiety, or maybe the combined effects of activating the GIP, GLP-1, and glucagon receptors as being more of the important drivers of the long-term weight loss. So that was kind of maybe a direction that was leaning after that. So now we get to the main phase 2 trial. So this was in people with type 2 diabetes. In this study, they enrolled 281 adults who were then randomly assigned to one of several different treatment groups. So some participants, they received placebo, some received dulaglutide, which we mentioned earlier, and this just served as this kind of active comparison, and the rest received one of 6 different retatrutide dosing regimens. And then the study then followed everyone for 36 weeks. Now, the reason here for randomization, it's really important because, it helps us make the treatment groups as similar as possible at the start of the study. So that just means in clinical trials that any differences that the researchers see by the end are much more likely to be due to the treatments themselves rather than differences between the participants. And, of course, these results still apply to the population that was actually studied. So this is type 2 diabetes. They don't automatically apply to every person with type 2 diabetes. And all of the active treatments in this study were given as a once-weekly subcutaneous injection. So the route of administration was consistent across the different treatment groups. And the study also reports 2 slightly different populations. There was this safety population, and it included all 281 participants who received study treatment. And it was basically used to analyze these adverse events that could happen. And then the efficacy population included 275 participants, and it was more used for analyses like changes in hemoglobin A1C as well as body weight. And just quickly, hemoglobin A1C, it's basically a marker of, diabetes, type 2 diabetes specifically, is commonly associated with your hemoglobin A1C. So I just want to bring that up. It's important in diabetes. And so when I throw around the term HbA1c, or you see that in the video, it's a marker for diabetes. And so this distinction is pretty common in clinical trials, this distinction between a safety population and an efficacy population, because not everyone who receives a treatment ends up contributing useful data for every efficacy analysis. So why that's important is because it's to pay attention to which population is being reported. So if the denominator differs slightly, it can differ slightly depending on whether you're looking at, the safety or the efficacy outcomes here. So that's kind of why we could have two. And before we look at the results, let's just take a quick look at the dosing, how the dosing worked for this phase 2 trial. So rather than immediately giving everyone this high dose of retatrutide, the investigators used this thing called dose escalation. And so basically all that is the participants, they started at this lower dose, and then, this dose was gradually stepped up over time until they reached their assigned maintenance dose. And the study included 5 different escalation schedules leading to maintenance doses of 4, 8, or 12 milligrams, as well as a fixed 0.5 milligram group. And then placebo and dulaglutide 1.5 milligrams served as the comparison groups. And one thing to note here is that while the paper reports the order of the dose escalations, it doesn't actually provide the exact timing of every step up, so we can confidently show the dose sequences without inventing the specific calendar weeks. So the main idea here is simply that participants were gradually titrated up to improve tolerability before reaching their target dose. And the other important point here is how the data were analyzed. So, rather than simply just averaging everyone's final measurements, the researchers used this statistical method called MMRM. This was mixed models for repeated measures. And, it sounds complicated, but the basic idea, it's pretty intuitive. So instead of just looking at one measurement at the end of the study, MMRM, it uses all of the measurements collected over time from each participant, while also accounting for the fact that repeated measurements from the same person are obviously naturally related to one another. So the study also specified that if a participant needed rescue medication or permanently stopped treatment, any measurements collected after that point, they were actually excluded from the primary efficacy analysis. So when we look at the results in the next few slides here, I just want you to keep in mind that these aren't just simple averages of everyone who was randomized. They're actually estimates generated using the trial's pre-specified analysis plan here with MMRM. Okay, so now we're looking at the primary glycemic outcome. This was the change in hemoglobin A1c after 24 weeks of treatment. And so if we remember here, just hemoglobin A1c or HbA1c, it just reflects the average blood glucose over the previous 2 to 3 months. So lower values indicate better long-term glucose control. And each dot here that you see represents the average change for one treatment group. And the further left the dot is, the greater the reduction in hemoglobin A1c. And so placebo, we see there at the second from bottom, it basically produced essentially no change, while dulaglutide reduced HbA1c by about 1.4 percentage points. Looking at the retatrutide groups, you can see a clear dose-response relationship. So the lowest dose shown at the bottom, the lowest dose reduced HbA1c by about 0.4 percentage points, while the highest dose regimen reduced it by over 2 percentage points, exceeding the reduction seen with dulaglutide. And so the standard error, or SE, shown at the bottom, is just simply a measure of how precisely these group averages were estimated, and the reported values suggest reasonably precise estimates here. So the main takeaway from here is that retatrutide produced pretty substantial dose-dependent reductions in HbA1c, with the greatest reduction seen at these higher doses. Now, rather than looking at the average hemoglobin A1c reduction, this table looks at a clinical target. So specifically, it shows the percentage of participants whose hemoglobin A1c fell below 7%, which is actually a common treatment goal for many adults with type 2 diabetes. So if we look first at week 24, only 18% of participants in the placebo group reached that target, compared with 52% in the dulaglutide group. And so across the retatrutide groups, the response rates ranged from 32% at the lowest dose up to 88% in one of the higher dose regimens. So by week 36, those response rates remained high, with around 80% of participants in the highest dose retatrutide groups maintaining an HbA1c below 7%. So a message here is that higher doses of retatrutide enabled a much larger proportion of participants to reach the standard HbA1c treatment target than placebo, and generally more than dulaglutide as well. And so these percentages, they're the proportion of participants who reach the target in this clinical trial. Okay, so now we're looking at body weight measured at 36 weeks, and each point is the average percent change in body weight for one treatment group. And again, further to the left, the greater the weight loss, looking at the controls first. So we saw that placebo was associated with about 3% weight loss, while dulaglutide produced about 2%. And in contrast to that, retatrutide showed a clear dose-dependent effect here. So the lower dose regimens produced more modest reductions of around 3 to 10%, while the higher dose regimens, they all resulted in approximately 16 to 17% mean weight loss by week 36. So higher doses of retatrutide, they produced pretty substantial weight loss in this phase 2 diabetes trial, considering considerably greater than that seen with either placebo or the dulaglutide here. So before moving on, let's just look at how participants progressed through the study. So there were 281 participants in the safety population, 275 were included in this primary efficacy analyses, and 237 completed the full study, and 222 completed the planned treatment period. And then among the 190 participants who received retatrutide, 16 of those stopped because of an adverse event. And so this is just important because we also need to consider how well participants were able to stay on treatment. So if you're having some bad side effects and can't stay on treatment, that's also something to note here. And in terms of safety, the most common adverse effects here were gastrointestinal, so in your gut. And that's exactly what we'd expect with incretin-based therapies. And these occurred in around 35% of participants receiving retatrutide, compared with 13% on placebo and 35% on dulaglutide. So the overall incidence was very similar between the two active treatments here. And the study also reported 20 serious adverse events in 15 participants. And I just want you to keep in mind that, a serious adverse event, or SAE, is actually a regulatory term. And what it refers to is events like hospitalization or other medically significant outcomes, but it doesn't necessarily mean the treatment caused the event. So finally here, there were no deaths reported during the trial. And overall, the safety profile was pretty consistent with what we'd expect for this class of medications, although a study of this size, isn't quite large enough to reliably detect rare or long-term adverse effects. Okay. So now we move on to the phase 2 obesity trial. So we went over the diabetes trial. Now we're going to look at the obesity trial. And unlike the previous study, this one enrolled 338 adults with obesity or overweight with at least one weight-related medical condition. But importantly here, none of the participants had diabetes. They were then randomized to either a placebo or one of 6 different retatrutide dosing regimens, and it was all given as once-weekly subcutaneous injections. So this trial, it was designed to answer a different question, and that's how well does retatrutide promote weight loss in people without diabetes who have obesity or have overweight. And the study, it ran for 48 weeks with participants remaining on their assigned treatment throughout that period. And then it was followed by an additional 4-week safety follow-up after the treatment ended. And although treatment continued for 48 weeks, the primary endpoint here was assessed at week 24, where the researchers compared the percentage change in body weight from baseline. And I just want you to keep in mind here that as we go through the results, I want you to remember, or it's worth remembering here, that the dose used, as well as, the study population and the time point being reported, they're all important because the results were specific to those trial conditions and shouldn't automatically be generalized beyond them here. So now we're looking at the primary endpoint of the obesity trial, and that was the percentage change in body weight after 24 weeks. So let's start with placebo. So they lost about 1.6% of body weight over the first 24 weeks. And as the other treatment groups up here, they're all plotted on exactly the same scale so that we can compare how much additional weight loss was achieved with retatrutide. Okay, so now we can see all the treatment groups together, and there was this clear dose-response relationship. So the 1 milligram group lost an average of about 7.2% of body weight. The pooled 4 milligram groups lost about 12.9%. The pooled 8 milligram groups lost 17.3%, and the 12 milligram group lost 17.5%, compared with that 1.6% in the placebo group. So by week 24, Retatrutide had already produced substantial weight loss, with the higher dose groups approaching 17 to 18% mean weight loss. And, this only tells us, though, what happens at 24 weeks. It doesn't actually tell us whether those results were maintained over the longer term. So here we're looking at the average or mean weight loss at week 48 for each retatrutide dose. And so when I say mean, I just mean, no pun intended, that the average across everyone in the treatment group. Okay, so that's what I mean there. And at 1 milligram, the average weight loss was 8.7%. At the pooled 4 milligram dose, it was 17.1%. And when I say pooled here, I know I've mentioned it a bunch, it just means that the researchers combined participants from different dose escalation schedules into one analysis for that final dose. At the pooled 8 milligram dose, the average weight loss was 22.8%, and at 12 milligrams, it was 24.2%. So you can see that here, overall, the higher doses were associated with greater average weight loss by week 48. But I just want you to keep in mind that these are group averages, not necessarily a prediction of what any one individual person will experience here. Now, if we add the placebo group, which is that comparison group that received a treatment without the active drug, we can actually see their average weight loss was 2.1% at week 48. So that gives us this reference point for how much weight changed without retatrutide. And you can also see that the higher dose groups achieved substantially greater average weight loss than placebo. And so, the results are going to vary for an individual person, but if we're looking at the trial side results, this is what we have. So it doesn't establish, though, what happens after week 48. And so instead of looking at the average amount of weight loss, now we're looking at a different question, and we're looking at responder rates. So when I say responder, I just mean somebody who reaches a specific amount of weight loss. So here we're asking what percentage of participants lost at least 5% of their body weight, or at least 10%. And the participants were randomly assigned to receive either 4, 8, or 12 milligrams of retatrutide or placebo. And the study, again, is double blind here. So I'm just reiterating some of these things just to kind of give you an understanding of what's going on here from before. This is the same study. Now, by week 48, at least 5% weight loss was achieved by 92% of participants at 4 milligrams, and 100% at 8 milligrams, 100% at 12 milligrams, and 27% with placebo. For the at least 10% threshold, the responder rates were 75%, 91%, and 93% at 4, 8, and 12 milligrams, compared with 9% for placebo. So these responder thresholds, they give a bit of a different perspective from average weight loss by showing how many people reached these clinically meaningful milestones. And that final row then adds the harder 15% threshold and the placebo comparison, which we will now look at. So here we have the 15% group. So by week 48, 60% of participants receiving 4 milligrams reached that level, compared with 75% at 8 milligrams and 83% at 12 milligrams. And in the placebo group, the responder rate was 2%. So these results established how frequently participants reached each of these weight loss thresholds by week 48. But even these high responder rates don't establish whether the weight loss is maintained after treatment is stopped. Okay, so now we're shifting from the weight loss results to the study's participant completion, which is sometimes called disposition. And out of the 338 participants who were randomized, meaning they were assigned to the treatment group by chance, 78% of them completed treatment. And that's the overall completion rate we can confirm from the available data here. And there are also several pieces of information we don't have access to, including the completion rates for each individual treatment arm or group, and that's just the number of study visits completed, and several details about how missing data were handled. So we still have some uncertainty here, but we know that 78% completed the treatment. And now here we're looking at some of those missing details here. So we're looking at the hybrid estimand, and an estimand is basically the specific treatment effect that a clinical trial is designed to estimate. In a hybrid estimand, it defines one treatment effect question while also specifying different ways to handle different events that happen after participants are randomized, like if somebody stops treatment or misses follow-up. So in this case, though the exact hybrid estimand details, they were inaccessible, arm level completion rates visit counts and the percentages of participants who discontinued because of adverse events were also inaccessible. So none of those values can be reconstructed from the overall 78% completion figure alone here. Okay, so now let's look at the main safety findings from this phase 2 study. And the clearest pattern involved gastrointestinal or GI side effects. So it was basically the stomach as well as intestines. And these were dose related, meaning they became more common or more pronounced as the dose increased. And the other main pattern was heart rate. So heart rate increased, it reached a peak, and then declined over time. And the registry also reported one death in the 4 milligram start 4 group. And the registry is the official trial record that summarizes the key study information. However, it did not report the cause of death or whether the death was attributed to the treatment. So now let's look at the main safety findings from this phase 2 study. So the clearest pattern here, it involved the gastrointestinal or GI effects, basically just meaning it was the stomach and intestines that were involved. And then these appeared to be dose related, which means they just became more common or pronounced as the dose increased here. And the other main pattern was heart rate. So heart rate increased, it then reached a peak and then declined over time. So the registry also reported one death in the 4 milligram group, and that was the start 4 group. And the registry here is the official trial record, and that summarizes the key study information, but it didn't report the cause of death or whether the death was attributed to the treatment. So based on the available information, we can't determine whether the treatment was responsible. If anybody in the comments has any extra information on this, please let me know. It's very possible that I could have missed a small detail. This here adds the participant counts for serious adverse events across the registry arms. So in the order, the participant counts were 3, 3, 0, 2, 1, 2, and then 2. And then these numbers here, they describe how many participants experienced a serious adverse event in each treatment arm, but by themselves, they don't establish causality here. So now we're moving into a nested MRI substudy, and it was looking at metabolic dysfunction associated steatotic liver disease. It's also called MASLD or MASLD. And when I say nested, it's just a smaller study carried out within a larger clinical trial. And in this case here, the researchers selected 98 participants from the parent trial who had at least 10% liver fat at baseline. And then they measured liver fat using MRI-PDFF, which stands for magnetic resonance imaging proton density fat fraction. And this is basically a technique that estimates what percentage of the liver is made up of fat. So because only these 98 participants were included, the results from this sub-study apply to this imaging subgroup rather than to everyone who was randomized in the main trial. And this here shows how those 98 participants were distributed across the treatment groups, as well as how many had imaging available for analysis at week 24. So at baseline, the randomized counts were 19 for placebo, 20 for the 1 milligram group, 19 for the 4 milligram group, 22 for the 8 milligram group, and 18 for the 12 milligram group. Liver fat was then measured again at week 24 using MRI-PDFF, and week 24 scans were available for 14 participants in the placebo group, 16 in the 1 milligram group, 15 in the 4 milligram group, 17 in the 8 milligram group, and 15 in the 12 milligram group. So that's why the analyzed imaging data set is smaller than the number originally randomized here. Now, here are the MRI-PDFF results at week 24 for the participants in the MASLD imaging substudy. Now, in the placebo group, liver fat increased by 0.3%, and in contrast to that, liver fat decreased by 42.9% with 1 milligram, 57.0% with 4 milligrams, 81.4% with 8 milligrams, and 82.4% with 12 milligrams. So the higher dose groups showed larger reductions in MRI measured liver fat by week 24. But there is this evidence boundary I just want you to keep in mind. And MASH, which stands for metabolic dysfunction associated steatohepatitis, is this inflammatory form of metabolic liver disease. And MRI measured reductions in liver fat are kind of considered a surrogate outcome, meaning they act as kind of an indirect measure rather than directly measuring disease resolution. So these findings, they don't prove MASH resolution, improvement in fibrosis, which is scarring of the liver, or fewer long-term related events. But we do see this massive reduction in the high dose groups in liver fat here. And this adds another way to look at the MRI results by showing how many participants reached a liver fat level below 5% at week 24. And the responder rate was 0% with placebo, compared with 27% in the 1 milligram group, 52% in the 4 milligram group, 79% in the 8 milligram group, and 86% in the 12 milligram group. And you can also see that only a small number of participants had MRI scans available at week 48, with 8 in the placebo group, 9 in the 1 milligram group, 9 in the 4 milligram group, 8 in the 8 milligram group, and 9 in the 12 milligram group. And, this smaller week 48 sample should not be treated as missing percentages. So based on the information reported here, durability of the MRI findings can't be supplied. And, just like what we just talked about previously, these MRI results don't establish, biopsy outcomes or fibrosis outcomes. We're just looking at the relative liver fat change here. So this is a nested body composition substudy involving 189 participants from the larger trial, and body composition was measured using DEXA, which stands for dual energy X-ray absorptiometry. And DEXA uses low-dose X-rays to estimate how much of the body is fat mass and how much is lean soft tissue. And so lean soft tissue here, it includes things like muscle, but DEXA does not directly measure muscle strength or physical function. So the enrolled groups included 29 participants on placebo, 32 on 0.5 milligrams, 31 in the pooled 4 milligram group, 33 in the pooled 8 milligram group, 30 on 12 milligrams, and 34 on dulaglutide. And as pooled, that I mentioned before, pooled is just participants from more than one dosing schedule were combined into a single dose group for the analysis. I'm sorry for repeating myself. I think it's just important, though, to reiterate these things as we go. So these numbers are the enrolled counts, and they're not verified as the exact numbers included in the final analysis. Now, this panel here shows the average percentage change in fat mass from baseline to week 36 within each group. So fat mass fell by 4.5% with placebo and by 4.9% with 0.5 milligrams. It fell by 15.2% in the pooled 4 milligram group and by 26.1% in the pooled 8 milligram group. While in the 12 milligram group, fat mass fell by 23.2%, while the dulaglutide group had a 2.6% reduction. So the larger reductions here were seen in the higher dose retatrutide groups. But these are within group averages, and all that means is that each number describes the average change from baseline inside that group. So by itself, this here doesn't establish a direct between group treatment effect. And here, this is just adding the statistical interpretation to those fat mass results from the DEXA substudies. So, we had these results, and, on the left, we have our 189% person nested subset. And on the right now, we're looking at the placebo-adjusted difference, which compares each treatment directly against the placebo. So the values shown are with a 95% confidence interval, or you're going to see it as 95% CI. And so a confidence interval, it's just a range that shows the uncertainty around an estimated difference. So for the 0.5 milligram group, the placebo-adjusted difference was minus 0.4 percentage points with a 95% confidence interval from 4.0 to plus 3.2. And because that interval crosses 0, the data are compatible with no difference between the treatment and placebo. So now the rest of the placebo-adjusted differences have been added here. So for the pooled 4 milligram group, the difference from placebo was, minus 10.7 percentage points with a 95% confidence interval from minus 17.2 to minus 4.2. And for the pooled 8 milligram group, the difference was minus 21.6 percentage points with a 95% confidence interval from minus 27.1 to minus 16.1. And for the 12 milligram group, the difference was minus 18.7 percentage points with a 95% confidence interval from minus 25.1 to minus 12.3. So, here, unlike the 0.5 milligram group, all of these confidence intervals stay below 0, which means the data consistently support greater reductions in fat mass compared with the placebo. So this final bit here adds the interpretation and main cautions for these DEXA results. So on the right, we have this separation between fat mass from lean soft tissue. And I just want to reiterate here that lean soft tissue includes much more than just skeletal muscle. You know, it could be organs, water, connective tissue, blood, and maybe any other non-fat tissues here. So when we refer to DEXA lean soft tissue, just keep in mind it's not the same as muscle function or a proper kind of strength endpoint here. And so the box at the bottom kind of highlights some of these important limitations. So the analyzed counts, the estimand, the completion data, all these things, and the missing data methods, they weren't accessible here. So I just want you to keep that in mind, but this is some of the data that we have, and it is quite promising with Retatrutide. So now we're moving to the Transcend Type 2 Diabetes-1 trial. So this was a peer-reviewed phase 3 trial, meaning it's basically a large late-stage clinical study designed to evaluate how well a treatment works and how safe it is before potential regulatory approval. So the study enrolled 537 adults with early type 2 diabetes across 3 countries. The participants were randomized equally in a 1:1:1:1 ratio, which all that means is just each person had an equal chance of being assigned to whether the 4 milligram group, the 9 milligram group, the 12 milligram group, all for Retatrutide, or placebo. So it's all equal. And the treatments given, they were given as weekly injections with dose escalation up to the target dose, followed by a total treatment period of 40 weeks. So the primary endpoint here, which is just the main outcome the investigators plan to measure, was the change in hemoglobin A1C from baseline. And I'm going to keep harping on this, but this hemoglobin A1C, it's the measure of average blood glucose over about the previous 2 to 3 months. And then baseline is just the measurement before treatment began. And again, I'll be referring to this estimand here. And the estimand is just the precise treatment effect question that the trial is trying to answer, including how events such as stopping treatment or using a rescue therapy are handled here. And so here on the right, you can see 2 different analysis lenses, and they answer different questions. So the treatment regimen estimand, it follows outcomes under the assigned treatment experience. So this includes things like the trial's specified handling of treatment interruptions and rescue therapy. And when I say rescue therapy, I just mean an additional treatment that is given if a participant needs extra help controlling their condition. And then the other lens here is the efficacy estimand, which is that question, that hypothetical question that by focusing on participants while they remain adherent to treatment and before any rescue therapy. So even though both of these estimands come from the same trial, they're answering different questions. So that's why their numerical results can't be blended or directly combined. And this trial basically establishes a peer-reviewed comparison within the study, but it doesn't establish results for all populations with type 2 diabetes. So now we're looking at the main hemoglobin A1C results at week 40. On the left are the 4 treatment groups. It was 4 milligrams, 9, 12, and placebo. They're all given as once weekly injections. In the middle here, we have hemoglobin A1C, and it was measured in the central laboratory. And then on the right are something called the least squares mean, or LS mean, and the changes in hemoglobin A1C at week 40. And so an LS mean, or least squares mean, it's basically just this statistically adjusted average that accounts for factors included in the trial's analysis model. And looking at the results here, hemoglobin A1C decreased by 1.69 percentage points with 4 milligrams, 1.86 percentage points with 9 milligrams, and 1.94 percentage points with 12 milligrams, compared with 0.81 percentage points with placebo. And so, coming up here, we're going to discuss some of these treatment differences that'll be shown with the 95% confidence intervals. Okay, so for the confidence intervals, we have minus 1.18 to minus 0.59, minus 1.32 to minus 0.76, and minus 1.39 to minus 0.85. And the p-value is less than 0.0001 for all of them. And that's basically just this statistical measure that estimates how likely it is to observe results like these if there were actually no true treatment effect. Okay, so we see those differences there. So now we're looking at the body weight results using the peer-reviewed treatment regimen estimand. So the 4 groups, again, they received those once weekly injections of 4, 9, or 12 milligrams or placebo. And on the right, the graph shows the mean percentage change in body weight at week 40. So under this treatment regimen estimate, the mean weight loss was 11.5% at 4, 13.9% with 9, and 15.3% with 12 milligrams, compared with just 2.6% with placebo. So, these results, they do establish the weight changes observed under this specific treatment regimen estimate. And this here summarizes the weight responder analysis using these thresholds again. So instead of just looking at the average amount of weight loss, we're looking at the proportion of people who reached at least 5, 10, or 15% weight loss. Okay, so for at least 5% weight loss, roughly 75 to 85% of participants receiving retatrutide reached that threshold, compared with 26% receiving placebo. For the 10% weight loss one, it was 52 to 70% with retatrutide, compared with 7% with placebo. And for at least 15% weight loss, the range was 31 to 51% with retatrutide, compared with 3% with placebo. So these are the ranges we have there. And this here, it just summarizes the participant completion and some of the main limitations of the trial. So a total of 537 participants were randomized, and of those, 490 participants, or 91% of those folks, completed treatment through 40 weeks. And 504 participants, or 94%, completed the full 40-week study. And on the right here, we can see a summary of the reported safety outcomes. So treatment emergent adverse effects, or something called TEAEs, which are side effects reported after treatment begins, they occurred in 63% to 64% of participants across the retatrutide groups, compared with 57% with placebo. And so serious adverse events, or SAEs, occurred in 4%, 1%, and 4% of the 4, 9, and 12 milligram groups, compared with 1% with placebo. And adverse event discontinuation, which just means that participants stopped because of side effects, it occurred in 2%, 5%, and 5% of those retatrutide groups, compared with 0% with placebo. And there were 2 deaths in total. They were both in the 4 milligram group, and both were judged unrelated to treatment. There was no severe hypoglycemia or dangerously low blood glucose that was reported in any group. And we also look at dysesthesia, which just means an altered or unpleasant skin sensation, as this reported adverse event. So these reported GI events, dysesthesia, serious adverse events, and deaths describe safety over 40 weeks. So they're not establishing the risk of uncommon events or long-term safety here. So now we're moving to the TRIUMPH-1 trial. But before looking at the results, so let's just look at where this evidence comes from. So, at the time of this cutoff, the available evidence came from sponsor reports and Congress presentations. So it's not from a peer-reviewed publication yet. So that's why, I've labeled this slide as provisional. And on the left, you can also see that 2 different sources report slightly different participant counts. So the clinical trial registry listed 2,335 participants, while the sponsor reported 2,339 randomized participants. And so, rather than trying to reconcile these numbers or choosing one over the other, both source-specific counts, we're going to keep them visible because that's what each source reported here. And I just also want to note that most confidence intervals are unavailable in the public reports at this stage. So I'm just providing you with the information that I have available. And, if I missed anything, please do let me know in the comments, and I'll make a little comment that I'll pin, and I'll just make a correction there. Okay, so here we have the overall study design for TRIUMPH-1. The trial was registered as NCT 059-29066. And it compared retatrutide at 4, 9, and 12 milligrams with placebo over 80 weeks. Active treatment began at 2 milligrams, and the dose was increased every 4 weeks until the assigned target dose was reached. And the main endpoint or primary outcome the investigators planned to measure was the percent change in body weight from baseline to week 80. And I just want you to keep in mind the evidence boundary shown on the slide here is that at this cutoff, most confidence intervals and the complete analyzed participant counts weren't publicly available. So this just establishes the source-specific study design and reported participant counts. So this slide shows the sponsor-reported efficacy estimand results for body weight at week 80. So remember that the efficacy estimand, it answers this specific hypothetical treatment effect question by focusing on participants under its defined treatment experience rather than the treatment regimen approach we discussed earlier. So the reported mean percentage change in body weight from baseline was minus 19% with 4 milligrams, minus 25.9% with 9 milligrams, and minus 28.3% with 12 milligrams, compared with minus 2.2% with placebo. So this is just provisional results so far. And here we have, the reported point estimates here, and they're the single best numerical estimates of the treatment effect. But most confidence intervals are still unavailable here. So just keep that in mind. So this slide shows the sponsor-reported treatment regimen estimand results for body weight at week 80. So I just want you to remember that the treatment regimen estimand answers a different treatment effect question than the efficacy estimand. So it more reflects outcomes, under the assigned treatment experience, things like including the trial's specified handling under this. And so this slide, we can see some of the weight loss at the efficacy estimand for how many participants reached certain criteria. So 25% weight loss, 30% weight loss, and at least 35% weight loss. And we can see this dose response, again, with increasing doses and more individuals reaching their target compared with lower at baseline. And as for, this is just for provisional so far. And here we have some of the safety information. So, Triumph 1, they report at the time of this cutoff that there's some provisional results because they came from sponsor and Congress reports and had not yet been peer reviewed. But looking at the GI side effects, nausea was reported in 28.6% of participants receiving 4 milligrams, 38.4% with 9 milligrams, and 42.4% with 12. Compared with 14.8% with placebo. And then for diarrhea, it happened in 25.2%, 34.1%, and 32% across the 3 active doses, compared with 13.5% with placebo. And constipation was reported in 23.8%, 25.9%, and 26.1%, compared with 10.9% with placebo. And vomiting occurred in 10.6%, 22.8%, and 25.3%, compared with 4.8% on placebo. So there is some safety concerns here just with side effects. Now, this just adds a few more things that are maybe a little bit less common. So dysesthesia, which just means an altered or unpleasant skin sensation, was reported in 5.1% of participants receiving 4 milligrams, 12.3% with 9, 12.5% with 12 milligrams, compared with 0.9% with placebo. And then urinary tract infection, or UTI, was reported in 7.5%, 8.8%, and 8.4% across the active doses, compared with 5.3% with placebo. And then adverse event discontinuation, which just means that participants stopped treatment because of side effects, occurred in 4.1% with 4 milligrams, 6.9% with 9 milligrams, and 11.3% with 12 milligrams, compared with 4.9% with placebo. So these are the provisional side effects here. And now we're looking at a nested knee osteoarthritis group within the larger Triumph 1 trial. And knee osteoarthritis is the wear-related breakdown of the knee joint that can cause pain and reduced function. Pain was measured using WOMAC, which is a patient questionnaire that asks about osteoarthritis pain, stiffness, and physical function during everyday activities. The pain scale runs from 0, meaning no pain, to 10, meaning the worst imaginable pain. And from baseline, or the starting value, of 6, the reported improvements at week 80 was up to 4.3 points less pain, which corresponds to about a 73.1% reduction from the baseline. And this is also provisional. And now we're looking at the nested obstructive sleep apnea group within Triumph 1. And obstructive sleep apnea, or OSA as it's sometimes referred to, is basically this condition where the upper airway, it repeatedly narrows or closes during sleep, and it can interrupt your breathing. So what we're looking at here with the diagram, and we're looking at the kind of measurements used during a sleep study, like nasal airflow, chest movement, and blood oxygen. And the main measure here is the apnea hypopnea index, or AHI, and it just counts the number of complete breathing pauses and partial breathing reductions per hour of sleep. So the baseline AHI was 58.6 58.6 events per hour, and by week 80, the sponsor reported up to 36.1 fewer events per hour, which does correspond to a 60.6% reduction from baseline. So some interesting findings there. And now we're moving on to the 104-week TRIUMPH-1 extension study. So let's just take a look at who is actually included. So at week 80, participants had to meet several eligibility criteria here. So they needed a baseline body mass index, or BMI, of at least 35. They needed a BMI above 22 at week 80, and completion of week 80 while remaining on the study drug, and no treatment discontinuation or permanent dose reduction. And just quickly, BMI, it's a measure that relates body weight to height. And so from those who met these criteria, the first 532 eligible participants entered this extension study. So this was actually a selected group of participants who completed treatment and then tolerated it well. It wasn't a new randomization, all that just meaning that participants were not randomly reassigned into new study groups. So these findings are, again, provisional. And here we're looking at what happened after those 532 eligible participants entered the extension. So it continued for 24 more weeks, from week 80 to week 104, using these blinded treatment paths. And blinded, it just means the treatment assignments, they remain concealed according to the study design. So participants who originally received 4 milligrams, 9 milligrams, or 12 milligrams continued toward a maximum tolerated dose, or MTD, of 9 or 12 milligrams. And when I say maximum tolerated dose, it's, as the name suggests, it's just the highest dose a participant could continue based on the study protocol and individual tolerability there. And participants who had originally received placebo also switched to Retatrutide and likewise moved towards a maximum tolerated dose of 9 or 12 milligrams. So after the former placebo group switched to active treatment, there was no longer this clean long-term placebo comparison. So I just want you to keep that in mind as well. And here we're looking at the week 104 results from this selected TRIUMPH-1 extension cohort. And on the left are the efficacy estimand results. Now, across the 4 original treatment paths here, the reported mean percentage weight losses at week 104 were 27.9%, 29.5%, 30.3%, and 19.2%. And on the right are the treatment regimen estimand results, and they just reflect outcomes under the assigned treatment experience. So in the same original path order, the reported mean weight losses were 25.7%, 28.7%, 29.9%, and 18.9%. And so, this is not a clean long-term placebo comparison because the original placebo group had already switched to to active treatment. So just keep that in mind here. And this here just shows the main accounting and kind of interpretation boundaries for these extension results. So, although 532 participants entered the extension, the analyzed participant counts were unavailable. So we don't know exactly how many participants contributed to each reported estimate. And the previously carried count of 522 is therefore excluded and shouldn't be used here. So confidence intervals, disposition data, and some of this other stuff was unavailable. At least we don't have yet. We likely will when we get the peer-reviewed publication, likely in the appendices and throughout the article. But right now we don't have that stuff. So let's keep that in mind as well. So now we're moving to the TRIUMPH-2 trial. And like the recent TRIUMPH-1 data, these results are also provisional because at the time of this cutoff, they came from sponsor reports as well as Congress presentations rather than a peer-reviewed publication. So the study enrolled adults with type 2 diabetes and obesity and compared weekly injections of Retatrutide at 4, 9, and 12 milligrams with placebo over 80 weeks. And the sponsor reported 1,152 randomized participants. You can also see an earlier historical registry estimate of 1,000 participants, just as an aside there. And these two numbers, I'm just keeping them separate because they come from different sources, and I just don't want to silently kind of reconcile them here. And the trial is registered as NCT 0592979. So this is kind of where we are with this study right now. And here, in addition to this main trial, there's also a nested GSA2 group focused on obstructive sleep apnea. So we have this kind of nested smaller subgroup of participants within the larger trial. And at the time of this cutoff, the identity of the GSA2 subgroup was publicly known, but no public results had been released right by July 26th, which is the date of this recording, which is today. So, the absence of these results here don't tell us whether the findings would have been positive, negative, or neutral. We just don't have that data quite yet. And here we have the sponsor-reported efficacy estimate results for body weight at week 80 in Triumph 2. And so let's just keep in mind, again, provisional. And we see for weight loss, we see placebo at -4%, -12.7%, 19.1%, and 20.8% for our different dose escalation protocols compared to placebo at week 80. And for hemoglobin A1C changes, we see, again, another kind of dose response pattern here, but surprisingly, the 9 milligram group actually beat out the 12 milligram group with the hemoglobin A1C changes. But again, this is provisional data, and we'll likely get more data in the future here. So now we're looking at some common adverse events from Triumph 2. So let's take a peek. So for diarrhea and nausea, again, we see this dose response escalation. About a third of folks are having, diarrhea or nausea on Retatrutide at the 12 milligram group, and diarrhea a bit more consistent, closer to a third across all groups, as well as constipation. We're seeing roughly between 14 to 17% in our groups for constipation compared to 9.4% with placebo. We're seeing large decreases in appetite at the 12 milligram group and kind of less large decreases in appetite at the 4 milligram group. And with vomiting, we're seeing, again, a pretty clear dose response escalation with vomiting with Triumph 2 for these adverse events so far. And then for dysesthesia, we're seeing, again, another dose response escalation with participants in placebo having very, very little dysesthesia here. And UTI having, maybe some dose response UTI stuff going on there. But again, placebo still had a 6.6% UTI occurrence. So take that for what you will. We'll likely get some more data soon enough here. And here we're looking at discontinuation because of adverse events. So the reported discontinuation rates were 3.8% at 4 milligrams, 11.6% at 9 milligrams, 7.7% at 12 milligrams, and 4.9% in the placebo group. And so that's what we have so far for that. And now we're moving to the Triumph 3 trial. This study enrolled now 1,949 participants with a body mass index or BMI of at least 35, along with established cardiovascular disease, meaning they had already had diagnosed disease affecting the heart or blood vessels here going into the study. And participants were randomized in a 1:1:2 ratio to receive weekly injections of 9 milligrams of Retatrutide, 12 milligrams of Retatrutide, or placebo. And so they were randomized with double the amount of folks in placebo compared to what you'd see in the 9 or 12 milligram groups. And here we're looking at the week 80 body weight results. So the reported mean weight loss was 21.6% with 9 milligrams and 22.6% with 12 milligrams, compared with 3.2% with placebo. And so this is what we have so far with Triumph 3. And here we have cardiovascular events from Triumph 3. But again, we should also interpret these results cautiously. So MACE, or major adverse cardiovascular events, is just a combined outcome, and it groups serious cardiovascular events together. So the 5-component or MACE-5 definition, it includes more types of cardiovascular events than the 3-component definition we'll look at next. And then the graph shows the hazard ratio, or HR as it's listed on here, which just compares the rate of events over time between groups. So a hazard ratio of 1, or HR of 1, means there's no difference in event rates. And for the pre-specified in-study MACE-5 analysis, there were 44 pooled retatrutide events compared with 52 placebo events, and the reported hazard ratio was 0.82 with a 95% confidence interval from 0.55 to 1.22. And again, this 95% confidence interval, it's this range that reflects the uncertainty around an estimated result. And because this confidence interval crosses 1, the data do remain compatible with either lower or higher event rates. So the results don't establish either cardiovascular benefit or cardiovascular harm. And we also show this kind of separate non-pre-specified on treatment analysis with a hazard ratio of 0.73 and a 95% confidence interval from 0.47 to 1.12. So because this analysis was not pre-specified, it answers a different question than the pre-specified in study analysis. And, we shouldn't interpret it necessarily as the same estimate here, okay? So for the MACE-3 side, there was 3 events here. There was 27 pooled retatrutide events compared with 23 placebo events, and the reported hazard ratio here was 1.12 with a 95% confidence interval ranging from 0.64 to 1.96. And we have this separate non-pre-specified thing for a hazard ratio of 0.92 and a 95% confidence interval range from 0.51 to 1.65. So these 2 analyses, they do answer different questions, like I've mentioned earlier. And importantly, though, here, all 4 95% confidence intervals shown across the MACE-5 and MACE-3 analyses, they do cross 1, which means all of these estimates do remain imprecise. So these data only are like imprecise cardiovascular event estimates. They don't actually establish cardiovascular benefit or cardiovascular harm yet. And here we have the common adverse events that were publicly reported for TRIUMPH-3. So, take this for what you will. I'll just keep it up here for your reading. And this is the final discontinuation slide. So, 9.8% of folks who are on 9 milligrams stopped due to an adverse event, and 13.5% stopped who are on the 12 milligram group, compared with 4.8% on placebo. And now we're looking at TRIUMPH-4. And this is a separate clinical trial registered as NCT 059-31367, and it enrolled 445 adults with knee osteoarthritis who did not have diabetes. And like I mentioned earlier, knee OA, it's the wear-related breakdown of the knee joint that can cause some pain and reduced function. And participants were randomized in a 1:1:1 ratio, meaning they had an equal chance of being assigned to weekly injections of 9 milligrams of retatrutide, 12 milligrams of retatrutide, or placebo for this total treatment period of 68 weeks. And again, this is also provisional and ongoing. And here we have 2 co-primary endpoints. So we have 2 outcomes that are kind of equally important. So body weight is one, and the WOMAC questionnaire that we talked about earlier is one. And here we have those 2 endpoints. So we had body weight changes of 26.4% reductions at 9 milligrams from baseline, and 28.7 reductions from 12 milligrams from baseline. And then there was, on the WOMAC pain questionnaire, there was up to 4.5% points less pain on this questionnaire from baseline. And so that's a relative improvement in pain of about 75.8% from the baseline here. And for the treatment regimen estimand, for the percent body weight change, we see minus 23.7% at 12 milligrams and minus 20.0% at 9 milligrams. And for dysesthesia and stopped because of an adverse event, or SAEs, we have for dysesthesia around 8.8% at 9 milligrams, and 20.9% of folks at 12 milligrams had dysesthesia, and 12.2% of folks stopped at 9 milligrams, and 18.2% of folks stopped at 12 milligrams, compared to 4.0% at placebo. And interestingly enough, some individuals actually stopped here for what they perceived as excessive weight loss. So interesting there. So before comparing all these phase 3 trials, just to remember that, they're not a race. So even though each study is evaluating the same retatrutide molecule, each trial is its own separate randomized experiment. So every percentage has to be interpreted within the context of the specific study it came from. So yeah, just keep that in mind as we go forward here. So we have all these 4 different phase 3 studies here. So Transcend, type 2 diabetes-1. We have the peer-reviewed data from that. Triumph 1, we still have some provisional data from. Triumph 3 and Triumph 4. And these are all these phase 3 trials. And so, the participant populations are differing across these 4. So type 2 diabetes for Transcend, type 2 diabetes-1. Triumph 1 was obesity with no type 2 diabetes. Triumph 3 was individuals who had cardiovascular risk factors. And Triumph 4 was those with knee osteoarthritis. And also the times and the measurements of the time were different. So primary endpoints being at 40 weeks or 80 weeks or 68 weeks as well. And also the percent change in weight is not necessarily this shared axis for all these things that we're mentioning before. So different population, different duration. So just, yeah, that's important to know as these results come out later on. So this is not like a head-to-head trial right now. So now we're shifting from efficacy to safety here. So starting with the GI side effects. So the most commonly reported effects, they're nausea, diarrhea, constipation, and vomiting. And, one pattern we saw here in the studies is that these symptoms, they often occur during the dose escalation. So when we titrate up a dose or gradually increase the dose over time, so the body can adjust, some are able to continue treatment while others discontinue because of, GI side effects, whether it's diarrhea, nausea, constipation, or vomiting. So across the studies, recurring GI tolerability is this consistent pattern, but the actual kind of event rates, they remain specific to each individual trial and, shouldn't be treated as one universal incidence. And, likewise here, inaccessible phase 2 obesity discontinuation rates can't be invented or filled in where the data are unavailable here. And so here we have a highlight between 2 different safety signals that have appeared across the Retatrutide program, but we're not going to combine them into a single conclusion here. So just if you can try your best to keep these separate here, it's going to be helpful for when you're reading the data. And on the left is heart rate. I made a video on this, but in the phase 2 data, heart rate increased. It reached a peak around week 24 and then declined afterward. I have a whole video on this. So if you look up, is Retatrutide's heart rate increase dangerous? I made a whole video on it. And, this is an ongoing discussion with this cardiovascular trial that's ongoing. So, we'll probably get some more data in the next few years here from that. But on the right is dysesthesia, which is just that altered or unpleasant skin sensation, such as, tingling, burning, or abnormal feeling on the skin. In several of these phase 3 studies, they also reported dysesthesia, but each of these findings come from its own individual trial. And so, we're not mixing these together here. So yeah, it's important to know. And, the final risk summary here focuses on gallbladder events, pancreatic events, and how to interpret this serious adverse event reporting. So a serious adverse event is, not automatically the same thing as an event caused by the drug. So establishing causation, we need more information, like how the event was attributed, how many people were exposed, and the kind of complete event tables here. So that's why I'm just trying to emphasize, questions like attribution, whether the full tables are available. So just keep that in mind there. And then while we do have a readable serious event ledger from phase 2, the corresponding phase 3 serious adverse event and death ledgers, they weren't publicly available in full here. So there's a lot we still don't know. So yeah, it's just, we'll probably have some more results in the next year or two here. Okay, and another thing here is it has to do with evidence gaps that still remain. So on the left, we have DEXA. And so to determine muscle function, you would need direct measures like a muscle strength test rather than a body composition scan alone. So why this is important is because, if somebody's losing a ton of weight, and a lot of that weight comes off as lean soft tissue in the form of muscle, does the kind of, say, grip strength, which is a great surrogate for kind of, frailty, could decline. So if grip strength declines, that is, a risk. Like if there's too much muscle loss and you end up with a person who becomes sarcopenic, where basically you lose a lot of your functional muscle and it really impacts your function. So it'd be helpful to have, muscle strength testing going forward, as well as, looking at some of these other surrogate measures, like, weight, which we're doing here, which we are doing, but it's just important to continue to do that going forward. And it'll be important for all these studies to continue in, phase 4, even when Retatrutide does come to market, to look at long-term outcomes with all of this data here. And again, it's still investigational. There is a pretty large online component to this right now, whether it's people selling Retatrutide as, say, like RT30 or other things like that. But again, with some of this online stuff, you don't know necessarily what the identity is, the purity, the dose, or the sterility. And so sometimes folks will go to these third-party labs. And so there's a deep trust on the third-party lab that their testing is, quality and accurate. And so, there's all these kind of questions that come up with these different comparisons here. And so just to sum everything up, we talked about Retatrutide structure as this triple agonist. We talked about all 3 of the receptors. We talked about some of the preclinical evidence in a cell evidence with the receptors, as well as in the mice. We looked at phase 2 data. We looked at phase 3 data. We also looked at some of the long-term risks with Retatrutide. And with that, thanks so much for watching, and hope you have a great day.